My name's Christian Duff. I'm the facilitator tonight for this webinar session called Genetics for Reproduction, the Male Influence. I work for Southern Beef Technology Services, which most of you will be aware of, so I won't have to go through that. The presenter tonight for this session is David Johnston. We're very privileged to have David on the line again tonight. He did the session last week, which I'll talk about in a second. But David is the Senior Research Scientist for the Animal Genetics and Breeding Unit at the University of New England. He's also a major CRC research leader and did a lot of the research that went undertaken in the CRC over the many years. To take you through what we're going to be talking about tonight, and just talk a bit about the background behind this webinar session, this webinar series called The Cutting Edge. When we put this series together, we obviously were aware that the beef CRC was finishing up in June 30th, and they had many years of research that was undertaken, and particularly in the last phase of CRC was dedicated towards the genetics and breeding side of things. And we thought it was very important to get these messages out to industry with a particular focus on the bull breeders, and that's why we're running this series for you. As you can see on the screen there, this is the second series, the second session out of a course of 6. Last week we ran a session on genetics for reproduction, the female influence, and that was undertaken by David Johnston. This week's the male influence, so it's continuing on that line of genetics for reproduction, and this week we're focusing on the male. Just out of interest, if you're interested in looking at last week's session that was done on the 13th and you missed out on it, you can actually view that now from either YouTube. You can just search for that normal and find that, or you could actually go to our SBTS or TBDS website, go to the webinars page, and you can view it under the past webinars there. So I encourage you to look at that last webinar if you didn't get a chance to. So just to talk a bit about how we're running this webinar series. If you aren't aware, the webinar technology basically allows you to view the screen that the presenter is looking at. In this case, it'll be a PowerPoint presentation, and you can actually interact with the presenter. You can hear him speak, obviously, through your speakers in your computer, but you can interact with the presenter and myself via typing a question in the questions pane and basically submitting and sending that question. I'll be here to facilitate the questions, and we'll be pulling up periodically throughout the David's presentation to answer those questions and to make sure we interact with you. One thing we find is sometimes your question box or question pane disappears. If that's the case, then you just need to click on the double arrows to open it back up again, and you'll be able to find that. We also, if you're having audio troubles, and saying that, I probably shouldn't say this because you wouldn't be able to hear me anyway, but if you're having audio troubles, you can actually select to use telephone, and you can use that. You can see that in your control panel or your audio pane, and you can basically dial in a conference number and follow the prompts. But hopefully everyone's having good audio tonight, and you won't have to worry about that, but that's an option for you as well. So what we'll do now is, that's the housekeeping and the introduction for this series. I'll now hand over to David Johnston, who's actually going to give the presentation tonight. So this will just take a second to change presenters. If you just bear with us, we'll make that change.
Good evening, everybody. We seem to be having contact.
Yes, we can hear you, David.
Yes, good.
You just might have to accept that, yes. Yes,
good. Okay, good evening, everybody. Hope you've had a great day. Absolutely beautiful day here in Armidale. So for those that were with me last week, we're now going to continue on with the genetics of male reproduction. As Christian said, the questions will come during the talk. So if you're thinking of a question, just type it into Christian. We'll pull up halfway through or a couple of times through the talk, and that'll allow you to ask the question. So don't wait until we stop, otherwise you won't get it in time. If there's no questions, we'll just keep going, and there should be plenty of time to get through this talk. So genetics of male reproduction, my name's David Johnston. Obviously through the Beef CRC, a lot of this work is a huge collaboration, and in particular for this male work, Nick Corbett and Dick Holroyd were leading and responsible for a lot of the analyses and a lot of the experimental work. I designed the study from a genetics point of view and was heavily involved in the running of the project as well. So just in terms of what I want to do in this presentation, just a quick recap of last week in terms of the results from the female reproduction, because this is following on directly from that project in Northern Australia. I want to describe this CRC bull project, what we've actually done. Obviously, like last week, there's a massive amount of results, so I'm just going to pick out some key results on these new male reproduction traits, look at some of their heritabilities, that is the degree of genetic control, what are some of the relationships among these male traits, and then importantly, what's the relationship between these male traits to female reproduction. Just a little section on where we're going in terms of putting this all together, and then some take-home messages for bull breeders and bull buyers regarding this work. So just to recap on last week, the experiment that was run in Northern Australia, we had 2,200 cows at these different locations throughout Queensland, and as we saw last week, for those that weren't on, these cows have full reproduction history from puberty right through to 6 annual mating. So we took them through to rising 9-year-old cows, where these cows were measured for their ovarian activity and their reproductive tracts every 4 to 6 weeks throughout their life. And so we have and we know the pedigree on all these animals, so we have a massive amount of information regarding the genetics of these cows. So what we've now, and in terms of those cows, we know that there were low weaning rates in Northern Australia, but from that study, the genetic control of reproduction has a big influence, and we were able to establish component traits were highly heritable, particularly age at puberty and lactation and estrus interval. And that lactation and estrus interval captures a big bit of the genetic variation that exists for lifetime reproduction. And so these were large half-sib families, and we saw more genetic variation in the Brahman. So a big opportunity there to change these traits through selection. And through breeding, through measuring these traits, we can change them. But what we realized is these traits are difficult to measure in the fact that we have to record females. And that means when we're making selection decisions, there's that delay in getting the information. So the obvious thing is, are there any male genetic indicator traits? Can we measure things on the young bull that tells us genetically about the genes he carries for female reproduction? Now we have one of these at the moment in genetic evaluation, and that's scrotal size. It allows us to tell differences for scrotal in male progeny, or differences in male progeny. But most importantly, it's an indicator trait for female reproduction, and that is the days to calving EBV, where we know there's a genetic association between scrotal size. However, there's opportunity to use more novel traits, and this is what the CRC investigated, traits like sperm morphology, blood hormones, proteins in seminal plasma. I'm not going to talk about the last one, that's still ongoing work, but certainly there was, when the CRC commenced, there was this obvious question was, is there additional traits that we could be using over and above what we use with scrotal to estimate their genetic control through a well-designed genetic study, and then estimate the association with female reproduction. That is, can we measure genetic indicator traits on these young bulls and then select on those traits to improve female reproduction? So the CRC study, again, just very briefly, the project design, we had the 2 tropical genotypes, the purebred Brahman and the tropical composite, which were from the pastoral companies, which represents 50% adapted genes. The dams of these young bulls that we measured were from the reproduction studies that we talked about last week that were fully recorded. They were then naturally mated to industry sires, and several of those sires were used as link sires to generate genetic linkage across the different locations. And the crux of this experiment was that the bull calves were retained until 2 years of age, and they were extensively measured for these traits. And we did that at 2 locations, Brigalow and Belmont Research Station. So the calves were born on the 4 different locations and then transported to either Brigalow or remained at Belmont. So we had our 2,200 cows, 1,200 composites, 1,000 Brahmins. They were then mated over the course of the CRC over nearly 10 years to 136 different sires from tropical composites and also purebred Brahmins. The project generated 9,260 progeny. Now, some of those, we had some calf losses, but of those male progeny, then we retained 3,533 young bulls from 7 odd cohorts. So we didn't measure all the bulls. We measured up to the 2010 drop bulls, and these were retained on those 2 locations. So anyone out there who's a bull breeder would understand the logistics of running big, large groups of young bulls. From a management perspective, that's no easy feat. But from a research perspective, it also meant large numbers of bulls that we were measuring. And this is what I want to focus on tonight, is the measurement on these young bulls. So we're talking big numbers here and the sort of numbers you need for genetic studies. The measurement regime, so at 0 months of age, we got a birth weight. Then at 4, 6, 12, 9, 15, 18, 21 and 24 months of age, we measured weight. We repeat measured scrotals. We took blood measurements at 4 months at branding and 6 months at weaning, and then 12, 18, 24. And then BBSE is bull breeding soundness evaluation, which I'll talk about in a minute. We performed on all bulls at 12, 18 and 24 months. So a very extensive recording strategy and repeat measurements of these traits. In terms of the measurements, so each of the bulls, when he came in at those different measurement time, we measured scrotal, we took blood for hormone work, then we used electroejaculation to get a sample, a semen sample from these bulls. Then we got a crush site assessment of that, but we also then took a sample and sent it off to the laboratory for morphology work. In terms of the hormone measures, we measured LH, or luteinizing hormone, which was after a GnRH challenge at 4 months, which we know that LH is associated with testosterone production and also influences the onset of puberty. We measured inhibin concentration at 4 months. It's produced by the testes and known to be part of regulation of sperm production. And we also did IGF-1, which is insulin-like growth factor 1, which is a hormone that we took after a blood spot card at 6 months of age at weaning. We know IGF is related to growth and the growth hormone axis, and also it's been shown to be associated with scrotal size and sperm morphology in bulls. And our CRC work showed that it was associated with heifer puberty. So these were candidate genes, or candidate hormones, that we thought were good candidates for measuring to see if they were good predictors of female reproduction. We also performed the breeding soundness evaluation, or the BBSE, and it does physical scores on legs, body condition score, feet and eyes, reproductive check of scrotal, testes, etc. Does a semen evaluation, which is a crush side, mass activity and motility, and I'll explain that a little bit more in a minute. And then importantly, it does a sperm assessment of morphology, looking at normal and abnormal sperm. Now this BBSE is an Australian cattle vet accredited scheme and provides certificates for bulls that are tested. The percent normal has been shown to be a phenotypic predictor of calf output from multiple sire mating, and therefore is an important candidate also for a trait that could be useful in genetic evaluation of female recording of reproduction. If you want to learn more about the BBSE, there's lots of information on the web, but the Future Beef website, which I've put up there, is a good link to learning more about the practical aspects of the BBSE. In terms of these semen measures, we took at 12, 18, 24 months. The mass or mass activity is a crush side score of the ejaculate, 40 times magnification, and simply a score of 0 non-active, 2, 3, 4, 5 up to rapid distinct swirl under crush side microscope. Progressive motility or MOT is crush side score again under higher magnification and scoring the ejaculate for percent motile sperm from 0 to 100. Motile sperm, yes. Percent normal sperm, this is where a sample is sent off to the lab. It's under 1,000 times magnification, and 100 spermatozoa are classified by an accredited morphologist where they're looking for 0 to 100% normal sperm, or how the percentage of normal sperm. So you can see each of those measures is ramping up to being, you know, a higher magnification and a greater degree of examination. So Christian, that's a quick description of the traits we recorded and the number of animals. So I don't know if there's any questions that have come in up to this point about the design or the animals, but I could stop now and quickly see if there's any questions at that point.
Yes, thank you, David. There's a few questions come in, but they're more related to the outcomes from research, I think, in this area. So they're not so much in regards to the project or design. So I might just hold off and ask those questions when we get to the next stage. But there's some good questions coming in, but I think it's more to do with the outcomes of the research rather than the project design. So we might leave that there. I will just add, though, we currently have about 103 attendees in, so it's another good attendance tonight, stretching from North Queensland all the way down to the Tasmania and across to the west. So thanks for everyone for coming in, and hopefully you're getting something out of it. But we might hold those few questions. Thanks for submitting those, by the way, the people who did, over to the next question section of this presentation.
Okay, thanks, Christian. I'm just going to now quickly go through the results. Before I get to the genetics, I just want to describe the data in a little bit of detail, just on a couple of quick graphs to give you a perspective on some of these measures. The first one is just looking at the live weights of these bulls from 6, 12, 18, 24 months of age. You can see the effect of the dry season here. These bulls were not heavily fed or supplemented. They were more under paddock conditions. They received no supplemental feed, and so these represents growth rates of bulls in the north. For a lot of people, these would seem low, but it's research where we also had all the bulls. Any bull that was weaned, we took through the experiment. There was no drafting, there was no culling, so it represents the entire calf drop of bulls from Brahmins and composites. So we see here at Belmont, where these bulls were directly compared, that at 6 months of age, 12 months of age, there's a slight difference between the composites and Brahmins. By 18 months of age and 2 years, they're pretty much very similar in their weights. Interestingly, their scrotal size, fairly similar at 6 months, but by 12 months, the two breeds are quite different now, with composites showing a much steeper growth of scrotal to over 25, 26 centimetres. By 18 months, there's considerable difference still, but by 24 months of age, the scrotal size in the two breeds was quite similar. And I think this is a key to looking at some of the results later and some of the results we saw last week in regard to the differences between these two genotypes in their reproductive performance. And I think this change in scrotal size here is very much demonstrating of that. If we also then look at some of the semen traits for motility, so this is percent motility on the left-hand axis, and age again across the bottom. You see here at 12 months of age, the Brahmins having about 15% motility, but in the composites on average about 40%. And then at 18 months, still quite a considerable difference, where the composites are nearly 60% motility, and by 24 months, both breeds are now up to 70% motility and very similar in their average performance. Likewise for percent normal, the measure of morphology, You can see at 18 months of age, Brahmins about 50%, composites about 65%. By 24 months of age, you see that both are about 75% average, percent normal. Once again, I think these differences between the breeds, we'll see a difference in how predictive these are then of the female reproductive performance. So if you can keep these graphs in mind or come back to them later, I think it helps explain some of the differences we see in the genetics of these traits and how they relate then to the female reproduction. Straight into heritability. So heritabilities explain the percentage of the trait that is genetic, and I've got here adjustments for weight and adjustments for age. Most traits we look on an age constant basis. People are out there interested to know how much scrotal is influenced by the weight of the bull itself, and if we adjust it for weight, what effect that has on the heritability. So here are the results for Brahmins, for scrotal measured at 6 months, repeat measured then at 12, 18 and 24 months. And you can see the heritability at 6 months is about 0.45, slightly lower weight adjusted, but then it goes to 0.65, 0.75, 0.75. So people who are familiar with heritabilities would immediately recognize that scrotal circumference is a highly heritable trait in this data set, probably higher than we see in other field data sets. Remembering that we're measuring all the bulls here of the complete drop, and the most genetic variation that we saw was at the 18 months in that dark blue shaded box, and the adjustment for weight was having no effect on the heritability estimate. So very comfortable that age adjusting is no different to if we were to weight adjust. In the tropical composites, again we see moderate to high heritabilities, 0.4 to 0.45 for scrotal circumference in our tropical composites, and at 12 months tended to be the time of measurement where we saw most genetic variation. So the largest amount of additive variance, for those that understand that, was at about 12 months of age. So just in summarising that, clearly scrotal size is heritable. The Brahmin estimates tend to be a little bit higher. Weight adjustment had no effect on our genetic expression, and most of the genetic variation, Brahmins at about 18 months, remembering the sort of weights we were getting in our experiment. These aren't heavy bulls by any means, but for tropical composites, it was the 12-month measure where we tended to see most variation. In terms of some of these other more novel traits, what did we see? In terms of the hormones, well, we have inhibin, LH and IGF-1. Again, moderate to highly heritable for these traits. I've mentioned scrotal there just to give it a benchmark. And then, for, some new estimates that people wouldn't be familiar with, semen quality traits, the mass activity, the motility, and the percent normal at 18 months. 18 months in Brahmins was also where we tended to see the most genetic variation for these semen quality traits, but we're seeing heritabilities of 0.25, 0.15, 0.25. So sort of range we see for growth type traits, we also saw for these semen quality traits. In terms of the tropical composites, the heritabilities of those bull traits, again for the hormone traits, inhibin, LH, IGF, moderate to highly heritable. So big differences between the sires in their sons, concentration of these hormones. Scrotal circumference 12 months 0.45, and again the semen quality traits for composites, because they're earlier sexual maturing in this data set, we see that at 12 months that's where we saw most of the genetic variation, and again heritability is 0.3 to 0.4. Very moderate to high correlations again for these new reproduction traits in our tropical composites. What sort of genetic relationship did we see between these traits? In terms of the hormones, what we saw was that IGF-1 tended to be moderately related to weight in Brahmins. So the measure of that growth hormone, that IGF-1, the insulin-like growth factor, is moderately related to the weight. So genetically the bigger animals had higher IGF. We saw that inhibin was moderately related to scrotal size. So we saw higher inhibin concentrations with those that were genetically bigger for scrotal. However, for LH, we saw no clear relationship across the male traits. So for some of the hormones and not for others, they tended to be related to weight and scrotal size. For scrotal size, we saw that they were moderately related to weight. So those with bigger scrotals were genetically bigger weights, and those correlations tended to be higher in the composites compared to the Brahmins. So composites, those correlations or those relatedness was stronger than in the Brahmins. What we did see was for scrotal was that they were highly related over the ages 12, 18, 24 months. They weren't quite as highly correlated for 6 months, although it was positive. We found that 6-month measure was difficult to get, particularly in the Brahmins standing in a crush. You know, there was a lot more measurement error in trying to get those measurements in those younger bulls. We did see between scrotal size moderate to high correlations with those semen traits. So the bulls with bigger scrotal size genetically were the ones that had higher motility, mass, and percent normal. So we start to get a picture now of the genes controlling these traits and the usefulness of scrotal size in measuring male reproduction. Finally, just a quick summary of the semen traits. What we found was mass activity and motility were genetically the same trait. So whilst they were scores under different magnification, at the genetic level, highly heritable and also highly genetically related. We see for each of those measures, mass and motility and percent normal, they were also highly genetically related over time. So a measurement at 18 months would rank the bulls very similarly as a measurement at 24 months, although the heritabilities did change over that time. And mass and motility were highly correlated with percent normal, but not 1. So they're clearly not the same trait, but they are correlated measures. So for these are all new results in terms of understanding the genetics of these male traits and understanding how these traits might fit into a genetic evaluation. So Christian, again, that's a very quick rundown of the heritabilities and genetic relationships of these male traits. If there was any questions pertinent to some of those, we could take them now, because what I go on now is just giving some quick results on how these results, how these traits were correlated with the female.
Right, there's quite a few questions about that in regards to how do these male traits correlate with the female traits, which is obviously the place to go. So I'll hold any of those particular questions over until the next question session if they haven't been answered. There is a couple here I'll throw at you now, David. The first one is a relatively straightforward question, but one we get from the field quite a bit. Is there a maximum scrotal size after which scrotal circumference is meaningless? For example, is 46 centimetres always better than 38, I guess, in regards to the genetic level? What's your comments there?
Yeah, well, that is a common question because, as we'll see later, scrotal has 2 roles. Scrotal is important to the bull himself as he stands in front of you, but then we're using scrotal as a predictor of his future progeny, his future sons, and his, in this case, also his daughters. So I'll mention this later, but the physical scrotal of the bull himself is important as a predictor of his likely fertility. And in different breeds, there is different relationships between the age and the weight of the bull, and there are different measures of what a minimum sufficient scrotal is to have adequate semen quality and semen producing ability. And in that case, you know, some breeds are using a cutoff of 32 centimetres at 400 days, but in other breeds 34 or 36. And in a lot of cases, the research would tell us once they're over that threshold, then having larger and larger scrotals physically as a bull isn't necessarily indicator of greater fertility of that bull as he stands there. When we're talking about the genetics, We're not necessarily talking about bulls that are 46 or whatever. We're talking about at 12 to 14 months of age, what is the difference between contemporary raised bulls, and in that case it's the differences that we use to then drive the EBV, where in the contemporaries the larger scrotal would be genetically correlated with greater reproduction in his progeny.
Thanks, David. There's also a couple of questions along the same lines, basically being there's out of the Brahmins and the tropical composites, there's quite a lot of variation there and some quite interesting correlations and heritabilities. How comfortable would it be to extrapolate that to, say, Bos Taurus, Cattle, Angus, Shorthorn, etc. in the south?
Yes, I'd be very comfortable in saying that we will have similar levels of heritability in our temperate breeds. What I'll show a little bit later is what the difference between Brahman and tropical composite would also suggest to me that in our Bos Taurus breeds, from a genetic point of view, in terms of using scrotal, I suspect we need to be measuring scrotal sizes at a younger age. So whilst breed plan has a window on scrotal size from 300 to 700 days, I'm starting to see from this research that if we want scrotal to be predictive in temperate breeds, we probably need to be taking scrotals at the younger end of that spectrum, and I've even got some research underway that suggests that that we will collect data even younger than that in Angus and Hereford bulls to see whether we should actually be measuring scrotal at a younger age than we currently do.
Good. Okay. The next question is quite a practical one that you may cover later on, but we'll answer it now and you can pass it off if you like. But basically, in regards to the semen traits, is there an appropriate age to collect that semen information, particularly when we're considering about putting that into an estimated breeding value potentially down the track?
Yeah, that's a good question, because that's always in the back of my mind. Where do we go from the research to putting it into practice? We see from this research that age does change the heritability of these traits. What we have to establish is there an optimum age for the genetic correlation with the females? I'll talk a little bit about that in the future, in the next couple of slides. But in saying that, age in this experiment is somewhat also dictated by what the weight of those bulls were, and that's what we have to look at before we make very clear recommendations to industry. Is it important when they're at a particular weight or a particular age? And given the influence of the wet season, dry season in Northern Australia, it looks like it's that measurement, probably in composites at about 12 months, but then in Brahmins after their first post-weaning dry season. So we've got to look at the timing, because it also has to fit with management as to when these traits are likely to be recorded, and I'll mention that in respect to with respect to the BBSE examination as well. So we might hold that one a little bit too, Christian.
Sure. Okay. This question's just a fairly broad genetic question, but it's you've mentioned it a couple of times in this last section of your presentation about the heritability of a trait changing with the age you collect that trait. Can you just comment on why the heritability would change, would go up or down based on the age you take the trait of the animal?
Yeah, the heritability might change with the age because different genes are involved as that animal ages. So some of the genes might be being switched on and some genes might be being switched off. Whilst we call it a trait, it's just a measure. So we weigh an animal. But a weight of an animal at birth compared to a weight of an animal at weaning or a weight of an animal at maturity, obviously there's different genes that can be turned on and turned off. And so the genetic architecture of that trait can be changing as an animal gets older. It can also change its heritability because the precision of the measure changes. So we in some aspects we might have more noise in our measures, well that will reduce the heritability. So it can be combinations of the precision and also the genetic architecture that can be changing the heritability of that trait over time. And also it can be, the change in the environment that the animal is under is changing the expression of those traits as well. And so, pre-weaning, we've got a particular maternal environment, post-weaning, etc. And so there are different genes being involved there that we petition out those effects for.
Thanks, David. I think we might hold the questions there now. We've got a few more, but I think a lot of them will be answered in this next section. So we might get back to your next section of your presentation, David.
Okay, good. So we've been through the male traits, what their heritabilities are, and their relationship with each other. So certainly they're heritable and they're related. What we now wanted to establish was, are they related to the female reproduction? And this is where our experimental design allowed us to estimate these correlations with the large bank of data that's been recorded on the females in this experiment. So we're able to determine what are those genetic relationships, and I'm just going to step through them. Obviously, I don't want to give lots of numbers. I just want to try and explain what the overall key results were. So I'm just going to go through the bull traits with the heifer puberty traits. So breaking up the female traits into a couple of different blocks. So the first one here is, what is the genetic relationship of the male to female? So that's saying, what is the relationship between the genes that we see in the bulls, and what is the relationship with the genes then we see in the female? Are they related? Are the genes associated with bull traits also controlling heifer traits? And so the first one is heifer puberty, and I've just put some sort of three dot points for each of these different slides. So the first thing we see is bull IGF-1 is a useful measure of heifer puberty. So this backs up our previous work with IGF-1 in the heifer predicting puberty. We see it again here that the bull's IGF is genetic related to heifer puberty. So the IGF measure taken in the young bull at 6 months is a predictor of heifer puberty. The next point is, we see that scrotal is a useful measure of predicting puberty in the heifer. We see that it tends to be the early measure, the 12-month in Brahman, and the 6-month measure in composite, so in the tropical composite. So it's as the scrotal circumference was taken later, so it relates just to that question we had. The later the scrotal, the less predictive it became of differences in heifer puberty. And finally, the semen measures, so the morphology and the crush site assessment of motility and mass activity were also useful measures in Brahmins having moderate genetic relationships. So that's the first one. The next one is, last week we focused on that first lactation cow. That's where a lot of the genetic variation existed for the female reproduction, the anestrus interval, and something like weaning rate in those first lactation cows. So if we look at those traits with the bull traits, we see that luteinizing hormone, or LH, has a moderate relationship in tropical composite, so one of the hormone measures. The scrotal tended not to be very consistent in its predictiveness of the lactation anestrus interval, whereas in Brahman the relationship tended to be positive with weaning rate. So immediately we see that scrotal was predictive of puberty, but not highly predictive of these cows coming back into cycling. Whereas we did see the semen traits were moderate to very high in predicting that lactation anestrus interval and the weaning rate. So we start to see a difference now between the predictive power, or the genetic predictiveness of scrotal versus some of these new measures, where the semen traits on those young bulls were predictive of the genetic differences in first lactation cows. So that's a really key result from this work. If we then look at the bull traits with cow lifetime reproduction, so these cows were measured through to 6 calving opportunities. Now those lifetime traits were lowly heritable. They're commonly sort of 7 to 10 to 15%. These yes, no, 0, 1 type traits, did you calve or not, often have lower heritabilities, and therefore these estimates clearly have less precision. There's more error, but what we saw was that the hormone measures generally were not consistent. Scrotal had a limited use, as in there were no consistent correlations. Brahmins at least tended to be positive, and the semen traits were very useful in Brahmins. So they had the semen traits were again predictive of lifetime, because that lactation anestrus in that first cow was so much important in the lifetime, and we saw that the semen traits were predictive of that. In the composites, the semen traits, morphology, so percent normal, and particularly mass and motility, were also positive in the composites, but of lower magnitude, therefore lower predictive ability. So just to finish up, what are we seeing out of the CRC before I go on to where are we going? The male traits are highly heritable, particularly the 18-month measure in Brahmins, the 12-month measure in composites. Scrotal is a useful measure of puberty if we're measuring it young, and that relates to the question about temperate breeds. But scrotal not much use, if any, for some of these other female traits. Some of the hormones have potential, particularly for puberty, like IGF-1, and it appears that these semen attributes measured crush side or with the morphology test are very useful genetic predictors. So that's a really quick snapshot of the CRC. What I want to go on now is, where do we go from here? So I don't know, Christian, we're running pretty well to time, whether you want to field any of those questions or whether we go into where I think we take this from here.
Yeah, there's some good questions there, but I think it's probably best to put up where you think we should go from here, and then we can come back to the questions, which I think will tie nicely into that.
Okay, so we'll keep going. So where to from here? Where do we take these results now in terms of the CRC? Well, the first one is scrotal size. It's an easy measure. It's mainly associated with puberty in this work. There's an existing EBV, and that EBV, and we use scrotal as a correlated trait in the days to calving, the female reproduction EBV in breed plan. So to me, scrotal sits there as an easy measure that we must continue to do. We saw some promise from the hormones, but I see these as costly and potentially difficult to take, with the exception possibly maybe of the IGF-1 if we saw that puberty was an issue in a breed and we wanted to improve female puberty, age at puberty. To me, the semen traits are very promising. However, they require this bull breeding soundness evaluation to be done, and of course that has a cost, and we'd have to figure that into our breeding program. And we have to be sure from a genetic evaluation that we're getting complete cohorts of bulls, that we're not just testing the bulls that we've bought to put out with our cows. That from a genetic evaluation, we now have to see how would we get this information from industry herds. We're starting to collect data and collate data from herds that have been doing this bull breeding soundness to start seeing can we replicate these heritabilities and genetic relationships in field data. We've got to resolve this age at measurement, so we're using the field data that we're collecting and this CRC data, starting to look at how sensitive are the results to this age at measurement as we go forward in looking to develop a genetic evaluation for these semen traits to allow that to build into an EBV for female reproduction. So that's the job at hand now that we take over as part of our breed plan project, taking the results now from the CRC. So what we need is to be able to use these male measures to improve female reproduction. So bringing it back to where I started this talk, why do we want these male measures? Because we can measure them so much earlier in a young bull rather than having to wait for a bull to have daughters where we measure the reproduction. So we can capture that genetic variation in those young bulls if we get good correlated measures. And therefore we have genetic information on the young bulls at the time that we're making selection decisions. And therefore we can make faster genetic progress in changing female reproduction. And therefore we select the young bulls to improve the female daughter traits. And that's where we've got to go in building this into a genetic evaluation. But to do that, we've got to first develop the performance recording for these male reproduction traits, and build that then into an EBV. So currently we have the days to calving EBV. As you saw last week, we can start to capture the age at puberty in that maiden breeding. We can capture the lactation and estrus by recording those traits. But now you can see my third point is into that EBV we can add scrotal size and sperm traits. And on the bottom, Mike Goddard will be speaking in a couple of weeks where we can also look to start building in genomic information from DNA, all leading to a single EBV for improved reproduction. And that might be called something like a weaning rate EBV. It may continue to be called days to calving, but capturing all those different pieces of information so that we can describe those differences and do selection to improve the trait through replacement bulls. It's selecting the young bulls where we'll make improvements in female reproduction, and particularly if we can capture scrotal size but also these sperm and semen traits. So in terms of the bull breeder, what are some of the take-home messages there? And I say in any breed, tropical or temperate, we've got to be recording scrotal size, particularly in Brahmins. We saw that the advantages there and the heritability is there, but at the younger age is going to be more predictive, and you've got to submit that data to breed plan on all your young bulls before you cut them or before you cull them. We've got to be capturing that information. The bull breeder then needs to be selecting on the days to calving EBV to be improving female reproduction. The days to calving EBV will capture all the benefits that exist through the correlation with scrotal circumference. Use bulls with adequate scrotal size in the stud herd, and that is for his own reproduction. But in terms of his sons and daughters' reproduction, the EBV will be the best guide. And I can see now that, particularly in Northern Australia, I'm not so sure about Southern Australia, but the bull breeding soundness evaluation is a tool that the bull breeder can be using to provide information for his bull clients, but also now start collating this data in preparation for the future, where hopefully we see these results build into future EBVs. So bull breeders, I'm sure most people are doing it, but scrotal circumference is a very easy measure to take and could be feeding into the existing genetic evaluation. In terms of commercial producers, obviously buying bulls with adequate scrotal size, we talked about that with one of the questions. You can be using bull breeding soundness results in terms of percent normal when purchasing bulls as an additional predictor to the likely reproduction capacity of that bull. But in terms of him passing on those, it's buying bulls with reproduction EBVs that's critical, using the scrotal EBV and the days to calving EBV, and hopefully a new and improved EBV in the future. So commercial producers looking to improve reproduction or maintaining reproduction in the herd, there's the bull himself that's important, but also then the genes that he carries. So that's just about got it wrapped up, Christian. Like last week, if anyone has future questions or further questions, just email me. Hopefully I've answered some questions that people had held over from last week waiting for this talk, but just shoot me an email.
Good, thanks.
Before I finish up, I've just got one further slide. As Christian mentioned, the beef CRC is now finished. So finished June 30. There are some new projects that are continuing on in Northern Australia with this work. Our AGBU MLA project will certainly now build on this to improve our reproduction EBVs. And finally, as a little bit of a free advert, is that the Belmont Red females, which are sisters to all the bulls that have been used in this study, are now up for sale. So these are young 2007 to 2011 drop females, the daughters from the cows we talked about last week. And if you're interested, this is some of them here in a photograph I took recently up there of the females that'll be up for sale in the tropical composite. So if anyone wants to get hold of them, give Rob Young a call. So that's a bit of a free ad there. There's certainly some very valuable genetics that can be used. In finishing up, like last week, these projects are a massive work under the CRC banner, obviously funded by MLA, but a large amount of cooperation from both our owners of the cows, but also a large number of people who supplied bulls, and then the Queensland research stations where these bulls have been run, and then a big team of technicians, scientists, but the team in bold there certainly did the most of the work in this project, and is certainly a credit to them. So I'll finish up there, Christian. We've got a little bit of time to take some questions. I hope people enjoyed the talk. Thank you.
Yeah, thank you, David. Yes, we'll take quite a few questions now before handing the presenting back over to myself to finish up. Just one question. I think you may have answered this, but just clarify it potentially. An attendee here basically said, with all this information now coming in for the bull fertility traits and the female reproductive traits, how is it actually going to all fit together? Do we need some kind of selection index to make it make sense? But I think you answered that in a way, saying we need a weaning rate EBV where this all feeds in. Do you just want to comment on that?
Yep, we certainly at this stage don't envisage introducing new EBVs, yet there could be some value in a new male measure, but in terms of its predicting the female, we'll funnel this all through a single EBV. It still might be called days to calving, because that still may be the best measure, but we may choose to rebadge it and make it simpler for industry to understand, and maybe call it a weaning rate EBV as well. So we haven't decided on that, but certainly our answer will be we'll feed it all into a single EBV, very similar to an index, where we weight the information according to the genetic relationship that we've estimated.
Thanks, David. The next question here, I guess, relates to another bull trait, which I don't think has been mentioned tonight, but correct me if I'm wrong, is libido. Has libido actually been looked at anywhere in conjunction with the semen traits and the scrotal size traits, and how that fits into female reproduction? Was that collected in this research?
Yeah, that's a very good question. No, we didn't collect it. So what would have been really nice is to take these 3,500 bulls and mate them to cows. But obviously the logistics of doing that was just way beyond this experiment. So we don't have actually any measure to say are these bulls related to, are these traits related to libido? Is that a separate trait? And then further, what is the genetics of calf getting ability? That's beyond this research to do that, which we dearly would have loved. So other research has shown that libido is heritable. There are differences, but we don't have any measure of that in this experiment.
Okay. In relation to IGF, just going back to that one particular trait, it obviously showed it had some useful correlations with heifer puberty, etc. Could this be also because it's got a relationship with weight, as I'm assuming, meaning IGF is one way correlated with higher weight, and therefore that could be bringing in the heifer puberty side of things just through the additional weight side? Is there any comments on that? Can you hear me, David?
Oh yeah, certainly IGF and weight are related. So it is picking up that, but it's also picking up, I think, sort of energy balance and maturity, and we saw it in the heifer. Those that had higher IGF at the end of the wet season were predictive of puberty, and likewise in these bulls. So it is probably mediating itself through the growth axis, but it's not simply weight. It's more early or that sexual maturity that the IGF is picking up, or those differences.
Yep. This is quite a practical question, and I don't have a good answer for it off the top of my head, and it probably depends on the cattle vet, I'd suspect. But how long does an average BBSE test crush side take if they're also obviously collecting semen for crush side semen and also morphology. Any comments on that, David?
Well, Nick Corbett and Dick could give you the actual minutes, because I'm sure they know exactly how many bulls they got through. Obviously it requires electroejaculation, but that is very, very quick procedure. So yeah, I've been part of a crew. I can't give you an actual time, but it's certainly doable on large groups of bulls at pretty quick speed. You need someone sitting on a microscope to be doing the crush side, taking samples, so it requires extra people, but the physical time to do it is not very, very long at all.
Very good. And probably just the last question to finish off these for tonight. I guess it's the pertinent question to come out of this, but when will the modifications to the EBVs be made? So we've got this all-empowering for female reproductive EBV. I guess when's a hard one to answer, but the question's come to me, David, so I'll throw it to you.
Yep. Well, we're working on it now. The first job we're doing is assembling these industry data sets on the BBSE. So where several breeders have been recording this trait for the last 4 to 5 years, we're now starting to look at that data to see can we replicate these heritabilities. Is it related to days to calving in those data sets, so that we can get confidence that we can go ahead and build those, what the genetic evaluation. So it's ongoing, and there's a few stop-goes that will occur along that process, particularly with regard to the BBSE and getting that in. The heifer reproduction, building those heritabilities from last week in, that will happen over the next 6 months, particularly in our, with our tropical breeds. Where we go with our temperate breeds is reliant on starting to collect additional records from AI matings, so that we can look to see if we can utilise that data. So two different programs for the north and south, but certainly as part of our breed plan development project, both these pieces of research and development are underway.
Thanks, David. Well, that finishes up our questions for tonight. I think we've done a good job to answer most of those. If you could now hand the presenting role back to me, David, and everyone, this will just take a couple of seconds, so if you just bear with us, we do have a little bit to finish up tonight, so I'll get you to hand it now back to me, David.
That should have you, Christian.
Yes, that works well. You've got me. Thank you very much. Okay, everyone. As we did last week, if you sat in, and we apologise for doing this because it's probably the last thing you want to do, but we're actually going to run a poll now. We're going to run 2 polls, so if you can just take a second to answer these. I'll put the first poll up, which will come up in front of you now. And basically, if you can just spend some time answering this poll. The poll basically is up in front of you now. Did you find this webinar useful? Strongly agree, agree, unsure, disagree, strongly agree. So if you can spend some time answering that, I'll shut it off in a couple of seconds. Okay. I've got about 90% of people have voted in this poll, so thank you very much. I'll just close it off now, and I'll ask you a second poll. So just bear with me, and I'll open up this poll. And again, it'll up in front of you now. So based on this webinar tonight, genetics for reproduction, the male influence, are you more informed about the application of the outcomes from the beef CRC, how they'll fit within your enterprise? Do you strongly agree, agree, unsure, not applicable, disagree, or strongly disagree? Again, I'll give you a few more seconds to answer that, but your feedback's really appreciated and something we really like to see. Okay, we're close to 90% being have submitted a vote on this poll, so again, thank you very much, and I'll close it off in a second's time. Great. So just to finish up this webinar for tonight, obviously at the completion of this webinar, We'll be making this available for viewing from the SBTS and TBS website, and also we put it on YouTube now. So if you hear of anyone who hasn't seen it or you want to watch it again, then it's obviously available for you from our websites or from YouTube. If you'd like the series on a disc, because it's a bit easy to look at for some reason, your broadband speed or something, then just get in touch with us. We can put that CD series on a disc. Saying that, we won't do it until the end of the series, so we've got another 4 of these to go, so it won't happen until the end. And obviously, if you have any of your questions that have been unanswered or your questions come to you after this, then feel free to contact, obviously, David Johnston. He's been kind enough to give us his email address. But also, feel free to contact the technical officer for your breed through SBTS and TBTS, and those there are in front of the screen. So as you can see there, you know for your particular breed who your contact is. For the tropicals, it's Paul Williams, and for the southern breed, it's either Andrew with Herefords, myself, Gemma Wilkinson, Peter Parnell, or Alex McDonald, and you should feel very free to get in touch with us to discuss any of these things you heard tonight. It'd be very remiss of me also not to thank David at the conclusion of this presentation. David's done an outstanding job. He's covered 2 sessions, quite detailed presentations with a lot of research, trying to fit 2 sessions into basically 2 hours, but I think we've all learned a lot from it. But saying that, it sounds like there's more to come, and I look forward to hearing more as we get down the track with this, particularly with female reproduction. So I'd like to thank David on behalf of our team and on behalf of everyone who listened tonight, and we will now call the webinar to a close. Look forward to seeing you in next week's webinar, and we'll send information via email on that in regards to that. So good night, everyone, and hope to see you next week.